Good afternoon, my dears. And today you can hear a lecture that is supposed to be written on the 2nd of May, but because it was a holiday, it is given with some delay. But it is not so important because we still have some classes ahead before this topic, before we start studying this topic. Okay? So now the topic of this lecture is infection and infectious process. And this is a really very doctor's lecture because today we are going to discuss a lot of new terms that we're supposed to learn how to understand and maybe new understanding of how bacteria behave themselves or viruses behave themselves in a body and due to what uh, factors, due to what capabilities they are able to cause diseases in humans, and also what are the differences between pathogenic and non-pathogenic microbes. And also we will take a look at some epidemiological features of the diseases, and this is what will we start with. At the very first slide you can see the very simple chain of the infection, the interacting trade of casual factors of the diseases. These are the factors connected with the, agent, factors connected with the quality of the microbe, including virulence, infectivity, addictive qualities, and this, etc. So then the factors connected with the quality of the host, genetic susceptibility to particular diseases, then nutritional status, motivation, resiliency, and so on. And also the factors connected with the environmental features. Public health sanitation, this is a rather social feature than biological, right? Social context, availability of health care, the level of health care, and so on. And then in this triangle, when we see this triangle, we recall the fireman's mantra. Can you see here? To make a fire or to let fire go on, it requires the air, because without air, fire won't happen, a fuel, and the heat. So this is mantra of the microbiologist: environment, agent, and the host. Okay, so first we will introduce ourselves to some definitions or statements. So what do we understand under infection? Infection is really the lodgement and multiplication of a parasite within or on the host body, right? But then, from the microbiologist's point of view, we are all infected, right? My dears, take a look at our body. We have bacteria that are living outside the body on the surface of the skin. We have a lot of bacteria that live in the oropharynx. We have a lot of bacteria that reside in the gastrointestinal tract. So these parts of our body are permanently colonized with bacteria. In some sense, we are infected. So then we go for the infection process. What is it? So the process of infection involves interaction between the human or animal body, named the host, and the infecting microorganism. So to some extent, we are uh, in specific relationship with our microorganisms that reside in our body. But then you should understand that The infection as a result of infectious process may or may not result in infectious disease, right? So when the normal, not highly pathogenic microbe used to live as parasites in our body, most probably it won't result in the infectious disease. So what is the infectious disease? I think this is the infection process that comes to the disruption of the normal functioning of the host. That's when the relationship between host and microbe stop to be normal and led to the damage to the body. So, the infectious disease is the type of infection in which we may see clinical manifestation of the particular disease, right? So then, this is the border in between just some interaction between microbe and the host, and the process of interaction is called infectious process, and the final result of this infectious process when this interaction led to the tissue damage or clinical manifestation of the disease. So from that moment it starts to be evident that microbe is present and disease is going on. I hope you understand this. So now, Uh, that's why when we are talking about the phenomenon of the uh, clinical disease, including infectious disease, we use the phenomenon of the iceberg. Can you see here? This is the iceberg in a sea. So, clinical disease, including clinical infectious disease, is the top of the iceberg visible on the surface above that project above the surface of the sea, a marine surface, right? But then submerged portion is much larger, but then it is a hidden burden of the disease. That's the iceberg concept of the infectious disease and population, right? So we don't see the moment of the exposure without infection. Then infection without clinical illness. And then maybe even when the symptoms are very mild, we may not still see the disease. Then when it comes to more severe clinical symptoms, then it starts to be visible clinical severe disease, or what do we have at the tip of the iceberg? And this is the most pessimistic outcome of the clinical disease. This is the death, right? So, but then we may have a not so pessimistic outcome of the disease. For example, recovery, right? Then the person recovers. So, and then we have something in between death and uh, recovery. This is chronization of the process and development of the chronic long-term disease. We'll discuss it a bit later on. So now again, the chain of infection. See, in this simple one, we need a microorganism that we can see some germs here, right? So bacteria, viruses, parasites, whatever. Then what serve as a reservoir of infection? When we are talking about pathogenic microbes, mainly I would like to underline, not always, but mainly, they the germs live in the human being, right? In the animals, sometimes in the pets, but then in wild animals also possible. But then sometimes the microbes may reside in food contaminated by human or animal, in soil also contaminated by human and soil, and water also contaminated by human uh, excretion, for example, and animals. Then we have path from reservoir to the new host. So that's really the answer, how germs get out of the body or get out of the animal. And there are many ways. So we may excrete it through mouth with vomit or saliva, with cuts on the skin. If someone will come, see here, someone come in contact with the blood of the infected blood, right? Then during diapering and toilet stool, so we may catch it there. So because they will be excreted from someone. Then the mode of transmission. So how germs may get in, path of entry, how they are transmitted to the place of entry, right? So the transmission can be done by the direct contact, for example, by kissing, right? Or sleeping together, sexual intercourse, or by indirect contact through hands or toys or sand or whatever. Then it may be spread as airborne disease through air droplets, right? When we speak, sneeze, or cough. So this is the path of entry, the way of transmission. Then how it enters the body, how germs may enter the body. That's not so many. Through mouth, through cuts or skin abrasion, through eyes, through ears, through genital organs, for example, or mucosa, or it can be inoculated in deep into the tissues. And then uh, it gets into the host that is susceptible to the disease. So, what does it mean susceptible to the disease? It means that the people who may be infected and are not protected from this disease. For example, very commonly, so the when the pathogen is very common, we perform vaccination of the more often children, right? So then children start to be not susceptible to the disease. Even if the microbe enters, it will not be able to cause disease because the antibodies arranged in the non-susceptible host will down-regulate the multiplication of the microbe and won't allow it to multiply. That is the sense of vaccination, to make people not susceptible to the disease, right? But then, who is susceptible? Those babies, for example, immature babies that had not been vaccinated, for example, for hepatitis B, because this is the very first vaccination type of vaccine given in to newborn babies, right? If the baby is premature, we have contraindication for infection. Maybe the baby missed this infection, then the baby will be still susceptible to this disease. Okay? So the susceptible disease and susceptible people are those who had been not vaccinated. Then we have a lot of HIV positive individuals, right? So the immunity is depressed or suppressed. So even if we vaccinate them, they may not produce very good level of antibodies or immunity against the microbes. So they are always susceptible to most human common diseases, right? So, but generally, uh, anyone can be susceptible. For example, if we take the influenza, of course, we are all susceptible if we are not vaccinated properly. But here we may have also some variations. So we may have high risk groups, people who are susceptible more than the others. For example, for influenza, this is very young children and very older people, right? Because the older people, they, their immunity is suppressed already with aging, right? So the common infection may cause more severe complications and even death of the patient. And the same with newborn babies or very small babies that are more susceptible than the adults. Okay, so that's, and those who are infected again get the agent that start multiplication in a body. So new cycle starts. And then those individuals start excreting these microbes. So this is the chain of infection. So, my dears, when you will be not so occupied, you take a look at this because this chain on infection is also given regarding or referring to the coronavirus COVID-19. I won't pay very much attention on it right now. But then if you, when you take a look at the lecture, I know that a lot of students are not listening for a lecture in a moment when it is delivered, okay? So they listen when they are in a good mood, okay? So when you will be in a good mood, please take a look and read carefully this because that's important for our everyday life because for more than 2 years already we are living in the world of COVID-19 and this very small virus regulates our behavior and many things and we never thought it would happen with us, okay? So go ahead with the terms. So, because of the, again, epidemiological chain, could you see here? So, everything's supposed to start with the germs, with the microbes that cause diseases. So, microbes that may cause diseases can be classified into several categories regarding their lifestyle or ecological features, right? So, the one type is they can be saprophytes. These are three free living microorganisms like Bacillus subtilis, Bacillus cereus, uh, but they usually don't need any living hosts. But then when they enter some immunocompromised hosts, that in which the protective immunity is not so high. They may get opportunity to stay in a body and to cause disease, to multiply in the body and to cause disease. So that's why they may cause usually diseases that are called opportunistic diseases or opportunistic infections because they get opportunity to do this, to cause disease. Then we have a second group that are parasites. And of course, we are of greater concern about parasites because what are they? They are microorganisms that may grow in living or on living tissues. But parasites are not all equally important in medical microbiology. we can have a lot of microbes that can be considered commensals because they usually don't cause damage to the host. For us, this is normal flora microorganisms. So those that live in our body without causing any harm. And there are a lot of good. So I hope you like yogurt. If you take a look what is there in the yogurt. So you can see that we can see there lactobacillus casei or some other lactobacilli. So these are very good bacteria that colonize our gut and prevent the multiplication of pathogenic bacteria. So they are very good true commensals in our body. But then within those commensals, so within those microbes that live in our body, we may find the so-called facultative pathogens. What is it facultative? It means they are not pathogenic microbes generally, but then under some conditions, they may start to be pathogenic and produce disease. For example, uh, so if we have a bacterial disease, we start taking antibiotics. But except for bacteria, so we may have candida in our oropharynx, right? So then, if we inhibit the bacterial growth with a broad spectrum antibiotic treatment, then the many groups of bacteria will be depressed. But Candida, that is a fungus, will survive and moreover overgrow everywhere because it will not need to compete with bacteria for food and place for ecological niche, right? So as a result, the Candida that seems to be true commensal now causes quite severe infection, right? So, and then finally, the group of so-called frank or primary pathogens. What are they? These are the highly pathogenic bacteria. And without referring to the level of immunity, as soon as they enter the body, they usually cause disease. And what we will study in special microbiology next year regarding bacteria and viruses mainly are frank primary pathogens. Okay, so now we're supposed to take a look at the property of bacteria to cause disease. It means we are answering the question, what is the difference between commensals, for example, facultative pathogens, and frank pathogens? Why some can cause disease and some microbes cannot? We continue in a minute. Guys, I just get some water, okay? So now, we are going to discuss two important properties, that is, or characteristics of micropathogenicity and virulence. What is pathogenicity? It is the capacity of microbial species, my dears. I would like to underline that this property is attributed to particular species of bacteria. So, uh, attributed to the capacity of the microbial species to produce disease. And it depends on the ability to invade the host, to multiply there, and avoid being damaged by the host defense mechanism. How to give an example? For example, we may have Staphylococcus aureus and Staphylococcus epidermidis, two species, both are staphylococci. But we consider Staphylococcus aureus species not, pathogenic, and Staphylococcus epidermidis as non-pathogenic. Understood? Or you know very well, Vibrio cholera. This is highly pathogenic microbe. But if we take a look at the members of the same genus, Vibrio, we may have a lot of non-pathogenic Vibrios that live, for example, in sea waters. For example, Vibrio mechnikovi, right? Named after Ivan Mechnikov, the one who discovered macrophages, right? So then it is absolutely non-pathogenic for human being. So, but they are from the same genus. So then the species Vibrio cholera is considered to be pathogenic, and Vibrio mechnicae species is considered to be non-pathogenic. So the pathogenicity is found or indicated on the level of species. Then we have a second characteristic that is called virulence. The virulence refers to the ability of the microbial strain to produce disease of particular intensity. So, first, strain. So we measure it on the taxonomical level of the strain. I would like to remind you, maybe you've forgotten already, that the strain of bacteria or strain of virus, right? So strain of bacteria or virus is a particular microorganism that is isolated or recovered from particular source and have a particular name or number in the journal under which we keep it in a uh, laboratory, and it has a particular source of infection. For example, if you take a swab from the person and isolate Staphylococcus aureus, so we write Madam Irina Andronovskaya strain taken from Madam Irina Andronovskaya, strain of Staphylococcus aureus, identified with all its properties, gotten on the 7th of November, let's say 1999. So we know exactly the source of the microbe and when it had been taken or isolated. So we call it a strain, right? So, but then we also may isolate a strain from the patient and it will be also identified as Staphylococcus aureus, but the properties of it can be slightly different. For example, that Staphylococcus, uh, because it was able to cause disease, had a plasmid that uh, code for hemolytic activity or lecithinase. So the properties will be slightly different, but then the virulence of it will be much higher than the virulence uh, of my strain. Okay? So then organisms that are freshly discharged from the host tend to be more virulent than those from the carrier who shows no sign of the disease. If I showed no sign of the disease, maybe my Staphylococcus aureus was not highly virulent. But Staphylococcus aureus taken or recovered from the, let's say, mm, tonsillitis, the case of tonsillitis caused by Staphylococcus aureus, that one will be much more virulent, while they belong to the same species. They both considered to be pathogenic. Understood? I hope you understand. So then, the virulence of the strain is not constant. It may undergo spontaneous or induced variations. For example, if we subculture this strain in unfavorable conditions, we may cause a reduction of the virulence. So, for example, for viruses, it's typical to passage the virus through unsusceptible hosts and repeating mm, passages may cause the selection of the a virulent strain, right? Or regarding bacteria, we may culture bacteria on artificial media or under not favorable temperature, not very optimal temperature. And then the bacteria viruses start mutating and they may reduce their virulence. But we may also increase the virulence of the microbe. And for example, it can be increased by multiple passages in animals. And then when after the passages through the susceptible through the animals, it will be again given to the susceptible host, it starts to be much more virulent. So then, and also virulence is a quantitative measure of pathogenicity, and it is measured by the number of organisms required to cause disease. For example, okay, now first a table that we may that may help to distinguish virulence and pathogenicity. So first look at the definition. So virulence is a measurement of the ability to cause disease. So in fact, virulence is a measure of pathogenicity. And pathogenicity is a general ability to cause disease in the host organism. Then measurement. Violence can be represented by a quantitative measure. Pathogenicity is qualitative measure and used in explaining the harmfulness. So violence can be used to express the degree of harmfulness of a pathogen. And pathogenicity not much suitable to explain the degree of harmfulness of the pathogen. Let's take a closer look. How do we measure the virulence of the particular strain? We may have several indices, right? So, for example, lethal dose 50. So, this is the number of the microorganisms needed to kill half of the infected hosts. No, I mean, infected hosts are usually laboratory animals. For example, we may find a dose, for example, 100 Vibrio cholera organisms. And this dose is, it means that if these 100 microbes is injected into an animal, it will, into the 10 animals, it will cause disease or death in half of those animals, in 50%. Then it is called LD50. Then that is the best dose because it may allow to compare the virulence. For example, for one Vibrio cholera strain, the lethal dose is 100 microorganisms, 50%. LD50 is 100 microorganisms. And for another Vibrio cholera strain isolated from another source, the LD50 is not 100 but 200. So what does it mean? That the second strain is less virulent than the first strain because much more or twice more organisms are needed to cause 50% death of 50% of the hosts. Okay? Then minimal little dose. This is the minimal number of the organisms required to kill 95% of animals tested under the same conditions. So, but then sometimes the microorganism doesn't cause the death of the animal. So then in that case, we use the index that is called infectious dose. For example, ID50, infectious dose 50. This is the number of the organisms that are needed to cause infection in half of the hosts. And that happens quite often because quite a lot of human microbes don't cause deaths in animals. They may cause some symptoms development, but not the death exactly. And also we may use minimum infecting dose. This is the minimum number of the organisms required to produce clinical evidence of infection in 95% of the uh, tested animals. So, how to compare? For example, the infectious dose of Shigella is less than 100 organisms. It is highly virulent. An infectious dose of Salmonella is somewhere 100,000 organisms. Why it is so? Because it is less infectious. So much more bacteria are needed to cause salmonellosis than shigellosis. Okay, we go ahead. So now we concentrate on the question how bacteria cause disease. So today we mainly, and it is easier to understand, using bacteria as an example. So when we studied viruses, we pay attention to the important factors that uh, cause pathogenicity of viral diseases, of viral infections. Okay. So, virulence factors of bacteria are structural or physiological characteristics that help organisms to cause disease, and we may broadly subdivide them into several categories. And these categories coincide with the usually stages of infection in humans. So, what is the first stage? And the group of factors that are important within this stage. So first is the adhesion. So why adhesion is so important? Can you imagine when we inhale, for example, some bacteria? So in our body, there are a lot of mechanisms that are involved in the elimination of bacteria from the body. For example, sneezing and coughing with the mucus, with the secreting of mucus, bacteria will be trapped in this mucus and we cough them out or sneeze them out of the body. So they will be removed very simply. But then a lot of bacteria may have factors that allow them to dissolve the mucus and to adhere tightly to the epithelium, for example. So these are called adhesive factors. So when bacteria adhere to the mucous membranes or skin or muscles and cannot be removed from this place, then they may start multiplication. Okay? So that adhesive factors are very important. And that is really a first step of the infection in a body. Then uh, in for bacteria, it's not enough to just to stay attached to a surface, right? For them, it is important to multiply. So, and not only to multiply, but then the offsprings, the daughter cells, bacterial cells, should stay also attached to the same surface, right? So we used to say that bacteria start colonizing the particular surface, the particular tissues. So, and uh, in these colonization factors help. Then, uh, sometimes it is enough for bacteria to cause disease. But more often, bacteria need to invade the deeper tissues. For example, they may enter the subepithelial layers and multiply there and even spread in the whole body, disseminate in the body. This group of factors is called invasiveness, ability to invade the deeper tissues and to spread in a body. And the final one is aggressiveness. This ability of the microbe to withstand the host defense mechanisms, namely our immunity. So, but then I would like to add one more because some of, and that's particular for bacteria, some of bacteria may be toxigenic. They may produce not just some enzymes but real toxins that are very harmful to the human body. So we go ahead and take stage by stage, step by step, take a look at the different factors, uh, group of factors that help bacteria to cause diseases. If we are talking about bacteria, the major adhesive factor is the Fimbria opili. You can see them here. These are hair-like projections. Please don't confuse with the flagellum, because flagellum is the locomotion organ. It helps bacteria to move. So, by the way, mm, high motility of some bacteria are also considered as a virulence factor. For example, of Vibrio cholera. So, because it also interferes with the ability to reach the particular surfaces that's supposed to be colonized. Okay. So then, Fimbria, that's a very important. But then in adhesion, and we may have a special type of fimbria that are called adhesive pili. So they are virulence factors. So then, apart from fimbria pili, for example, if we have some microbe that is not pileated, without pili, it may still adhere to some surfaces with the help of the capsule. You can see here that the capsule is the very sticky substance, usually made up of polysaccharides or mucopolysaccharides that are very sticky. So, and with this, it may attach very tightly, adhere to the surfaces of um, the body. And so for bacteria, this is a capsule, and for viruses, can you see here? Do you remember? This is enveloped virus, and these are the projections on the surface of the enveloped viruses that are called glycoproteins that form spikes, right? Those spikes also show for adhesion or adsorption of the microbe on the surface of the host organisms. So these also some examples of adherence, right? So there's a Neisseria gonorrhea organism that adhere to the very tightly to the epithelial surface of the genital tract with the help of fimbria or pili, and also to the urinary tract epithelium. So, and this is example of the capsule of Staphylococcus. No, this, my dear, this is not Staphylococcus. This is Klebsiella pneumonia. So, shown here. But then the Staphylococcus epidermidis also may have a slim layer of the or the capsule that mediate the attachment of bacteria to the endothelium of heart valves, for example. Then colonization factors. So this is colonization is the growing of the microorganisms on epithelial surface or other host tissues. For colonization to occur after adherence, the pathogen must survive and reproduce itself despite the host defense mechanisms, right? And you can see here how the uh, colonization may occur. See, first the host surface is detected, right? Or detected, right? And then the due to the involvement of quorum sensing, virulence will be activated and colonization of the surface will be occur because as soon as um, we have a population of bacteria, they start exchanging with some signals. These are usually very short peptides, oligopeptides, that can be sensed by other bacteria in population and uh, may cause the rearrangement of genetic material and expression of new genes and showing a new factors or synthesizing of new proteins that may mediate the colonization. And here you can see the example of colonization of the tooth animal by normal flora oral cavity organism like streptococci, fusobacteria. So they initially adhere to the membrane and then they form the layer on the surface of the dental animal. And then finally, due to the high biochemical activity of these microorganisms that ferment sugars and produce lactic acid and some other acids, so they may cause caries as a result of this adhesion. And here you can see, for example, how many different types of bacteria may grow on the tooth animal and to which initially particular microbes like Streptococci C, they first adhere and then they help a lot of other bacteria to form the plaque on the surface of the tooth animal. And starting from streptococci usually that adhere with the slim layer, then a lot of other bacteria, Veillonella, Prevotella, Haemophilus, Propionibacteria, Actinomyces, and all the others. They are connected together may cause dental plaque formation. That is the dental plaque formation. And then the damage to the tooth animal and forming of the caries. That's a very good example. So then we take a look at the so-called invasiveness. That is the ability of the pathogen to spread in a host tissues. The invasive bacteria produce spreading lesions depending on the ability of the pathogen to secrete several, usually enzymes, that may play a role in pathogenesis. So quite often the invasiveness is connected with the ability to produce particular enzymes. And now we take a look at how those enzymes may work. So you can see bacteria. So first they colonize, adhere and colonize uh, the epithelial surface. But we have the hyaluronic acid, that cement between cells, and beneath the cell layer we may have collagen layer. My dears, do you remember your histology, your knowledge in histology? So then, bacteria may start producing enzymes that uh, interfere with the cement, cell cement with hyaluronic acid. So the enzyme is called hyaluronidase. This is extracellular enzyme or exoenzyme. What does it mean? Bacteria produce this enzyme and secrete to outside. So, and outside bacteria, this enzyme find its substrate and dissolve it, right? So, uh, the hyaluronidase make the ways for bacteria to enter deeper. And if they start producing enzyme that is called collagenase, then the collagen fibers will be disrupted and bacteria invade the deeper muscle tissues and the others. Then how they manage to invade the deeper tissues. Then we mentioned already the flagellum, right? Now you can see Helicobacter pylori and how it may invade the tissues. So usually contact with stomach acid keeps the mucin lining the epithelial cell layer in a strong gel-like state. See, this is gastric mucin gel, and this very quite hard and quite good protection for epithelial cells. But this consistency is impermeable to a lot of bacteria, including Helicobacter pylori. It gives very good protection. But then the bacterium start to be release an enzyme that is called urease, which neutralize the stomach acid and pH start to be much more alkaline. This causes the mucin to liquefy, and bacteria with the help of their flagella uh, introduce themselves into this mucin layer through it to reach the epithelial cells. Here we can see two uh, factors of invasiveness. The ability to produce enzyme urease that cause the alkalinization of the gastric mucin gel and liquefaction of it, and also motility caused by bunch of flagella here that introduce the microbe or allow the microbe to reach the epithelial cells through the liquefied mucin. Okay? I think this is something new for you. Then we come up to this next group of factors that is called aggressive factors. So, and that sometimes it is difficult to see the difference between the invasive factors and factors of aggressiveness, right? But then usually the aggressive factors, we mean that they give protection from some immune mechanisms. For example, coagulase accelerate the formation of a fibrin clot from fibrinogen. See, this is called coagulase. It causes coagulation of blood. And this clot may protect bacteria from being phagocytosed because in the gel, phagocytes are not active. So coagulase is really an aggressive factor. Some bacteria may produce an exoenzyme that is called leukocidin. This enzyme destroy neutrophils and macrophages. That is also a way how to overcome host defense mechanisms. Then some bacteria may produce fibrinolysin, the substance that uh, dissolve fibrin clot. And this is called streptokinase if it is produced by Streptococcus pyogenes, or staphylokinase if it is produced by Staphylococcus aureus. But then the function is same, to dissolve the fibrin clot, and the bacteria trapped in this fibrin clot can be free and free themselves and spread in the body. So here you can see how it works. See? So this is a pathogen that is introduced into the deeper tissues by inoculation, maybe by punching of some object, right? Okay. And then when pathogens multiply, they produce, can you see the very tiny blue dots, right? They produce the protein molecules that are called coagulases, right? Then these coagulases cause plasma to clot, right? So the macrophages from the blood cannot digest this bacteria here, right? But then after a while they are multiplying there, they start producing an enzyme that is called streptokinase that dissolve the fibrin clot and let them into the bloodstream and spread there. Okay? That's how they may spread in a body. So we have taken a look already at the hemolytic activity of the Staphylococcus aureus, right? All right. So, and it is associated to this hemolytic activity is associated with the ability to produce an exoenzyme that is called hemolysin or hemolysin, someone pronounce. So this is the enzyme that lyses erythrocytes. But then generally we um, um, distinguish two basic types of hemolysin. So this is alpha hemolysin, and it causes just partial breakdown of hemoglobin, modification of hemoglobin into methemoglobin. So that's why the greenish ring around the colon is produced. Can you see here? So it causes the changing of the color of the agar, blood agar, right? It turns green. So this usually is typical for streptococci. They are called uh, uh, strep viridan streptococci. Viridan, it means green because they are able to produce alpha hemolysin. But then we may have the much severe damage to the erythrocytes, usually produced by so-called beta hemolytic activity. When we have the completely breakdown of hemoglobin and erythrocytes that leave the clear, not pigmented, transparent zone around the colony. Normally, the blood agar is opaque because the cells are incorporated in agar, it is not transparent and red. So when erythrocytes and hemoglobin is broken down, so we can see the clear zones around the colonies. The colonies are seen right and clear halo around the colony. So the capsule of some bacteria also considered to be aggressive fact as well, because and that is the appearance of the capsule in uh, mm, in fluorescent assay, right? So and they prevent digestion by phagocytes because can you see bacteria very thin, but the capsule can be very thick. It is really not digestible by macrophages. and that is the capsule of the Streptococcus pneumonia, right? Antiphagocytic capsule. So then some bacteria are capable of so-called intracellular survival. And then uh, typical for bacteria that can be obligate intracellular parasites. What does it mean? Obligate intracellular parasites. It means these bacteria, like viruses, cannot multiply outside the host cell. At least two types of that type of bacteria, that kind of bacteria shown here, chlamydia and their intracellular survival. And then also rickettsia. This is a small rods seen by Jim Sustaining. And they occur inside the cell, so that's why they are not will be easily digested by phagocytes, for example, and so on. Then finally, we mentioned toxigenicity. It means bacteria are able to produce toxins. And usually we may classify toxins into two large groups. What are those groups? We may have exotoxins and endotoxins. And my dear, there's a big difference between these two groups of toxins. uh, Here we have a table for comparing of these two types of toxins. First, let's take a look at the name. Why endotoxins are called endotoxins? Endo, inside. I don't think why. We just okay. Let's take a look this first. So endotoxins usually are produced by gram-negative bacteria only, while exotoxins are produced by both gram-positive and gram-negative bacteria. Then let's take a look at the chemistry. So exotoxins are proteins. They are of polypeptide nature. It means this substance can be produced inside the cell and secreted to outside the cell. Mostly they are exotoxins, right? Released into the environment. While the endotoxin, why it is called endotoxin? It is a part of the cell wall of gram-negative bacteria. So it means it doesn't have polypeptide nature. It is of polysaccharide nature. It cannot be dissolved in water, right? So that's why it cannot be really secreted to outside. It is a part of the cell wall. So that's why it is found in gram-negative bacteria only. And the chemical structure is described as lipopolysaccharide. And the chemical structure of the exotoxins is described as polypeptide. So then, these two substances react differently on the heating. Lipopolysaccharide, it is not a protein, so it can be boiled and still it will save the structure of polysaccharide. Okay? So, but then if we treat a peptide or polypeptide with the heating, it appears that it is heat labile because it will be destroyed within 30 minutes at 60 degrees Celsius. Then, now how the expression of these proteins is regulated. It means when and under what conditions these substances are produced and who will regulate the production of it. If we take a look at the bacterial genomic structure, of course, if we are talking about cell wall, that is the mandatory structure of bacteria, and the production of the cell wall is regulated by bacterial chromosome, major genetic element. You can see it here, right? While the uh, exotoxins are extra substances. Maybe they are not needed within the normal life of bacteria, but they are important when bacteria multiply in the human body. So then, usually, the regulation of the exotoxin production is under plasmid or bacteriophage control. It means that the genes that code for these exotoxins are incorporated into the mobile genetic elements like plasmids or bacteriophages or so-called, do you remember, pathogenicity islands, when we have the large cassettes of alterating plasmids and bacteriophages usually, and transposons, right? So then regarding toxicity of these substances, usually exotoxins have very high toxicity and micrograms are enough sometimes to cause some effect in the body, while the endotoxins has very low toxicity. Up to milligrams are needed to cause some effect in the body. And uh, these toxins may cause different types of the diseases. So, with the release of exotoxins, tetanus, botulism, and diphtheria diseases are connected, right? They are produced just because of the ability of the bacteria to produce toxins. While the disease is caused by endotoxin, there's like meningococcal meningitis or meningococcemia. This is just because the presence and autolysis or lysis over the gram-negative bacteria in the bloodstream. And when the bacteria, gram-negative bacteria are lysed, then the lipopolysaccharide and its most toxic component lipid A start to be exposed to the human tissues and human body fluids. So then it causes some toxicity. So and now a few words about the clinical effects. Uh, Usually exotoxins have very specific effect and particular for each toxin. For example, Vibrio cholera causes diarrhea, right? Watery diarrhea. And botulinum toxin causes the arrest of acetylcholine release. I think you know physiology, normal physiology enough already to understand my examples, right? So, but then the effect of endotoxins are not very specific. In fact, endotoxins of all gram-negative bacteria act in the same way. But regarding the different types of gram-negative bacteria, the effect can be more severe or less severe, but it is in the mechanism it is same. Usually, the release of endotoxin causes fever, very high inflammatory response, and sometimes, if it happens in the bloodstream, it may cause endotoxic shock. So then the property of tissue affinity, of course, because exotoxins have the particular specific effects, so they have a high specificity for the particular tissues. Why endotoxins act not specifically, so they won't act specifically. Then mode of action. Exotoxins are usually enzymes. Quite often they are enzymes. Endotoxin has no enzymatic activity, but it is a very strong macrophage activator. So under LPS action, macrophages that engulf gram-negative bacteria usually in order to kill them. They start to be activated and produce very powerful substances that are called interleukin-1 and tumor necrosis factor that are very strong so-called pro-inflammatory uh, interleukins and cytokines and may cause pyrogenic effect in the body. So it means the elevation, rising of the temperature dramatically up to 40 degrees Celsius and very strong inflammatory response. Then now the question is, can our body produce something to protect us from these toxins? Regarding the exotoxin, it is highly antigenic and usually the antibodies can be uh, induced in a body that are called antitoxic antibodies or antitoxins. And if they are produced, you can see the function of these antibodies can be to neutralize the activity of the exotoxin. So if we are talking about endotoxin, it is fully antigenic. Of course, some antibodies will be produced, but they cannot neutralize the whole bacteria. So that's why these antibodies are not so effective. And if antibodies are not so effective, no vaccines really available that will be very effective against endotoxin. But if we are talking about exotoxin, the mm, vaccine can be produced. But in fact, the exotoxins themselves cannot be used as antigens for vaccination. Why? Because they are very toxic. Can you imagine if we start using even minute amount of exotoxin for vaccination? It may cause very severe damage to the human body. So it means that we should do something with the exotoxin uh, to let it be not so toxic, but retain immunogenicity and antigenicity. And this is detoxification of the toxin, and the resulting substance will be called toxoid. So so what is the relationship between exotoxin and toxoid? Toxoid is not toxic exotoxin. Understood? So now you see, endotoxin is the upper part of the cell wall of gram-negative bacteria. See, lipopolysaccharide. It consists of all somatic antigen, uh, polysaccharide backbone, and lipid A that is a toxic component. As soon as it is ruptured, lipid A start to be exposed to a body. So, and the effect produced by endotoxin are fever, leukopenia, activation of the complement, thrombocytopenia, possibly disseminated intravascular coagulation where severe state. So it may decrease the peripheral circulation and perfusion to major organs. Then hypo, uh, uh, very severe hypotension, increased permeability of the blood vessels, hypotension, shock, and death. So, if we are talking about virulence factor and the toxigenicity, I would like you to take a look at the special systems developed in bacteria to deliver the toxin from inside the bacterial cell to the host cell. So you can see here that there are several types of uh, secretory systems associated with the special fimbria, right? So you can see here that type 1, type 3, and type 4 secretory systems transport the protein from bacteria to the host cell in one step from cytosol of bacteria directly to the cytosol of the host cell, out of the cell, right? So there's a very important system to deliver toxins inside the host cell. In the case of type 3 and type 4, the protein are transported from bacterial cytoplasm to target cytosol directly. So type 2 and type 5, type 2 and type 5 secretory systems transport proteins in two-step process. So first to the periplasm via the Secotat system before reaching the cell surface, and then to the cell surface. Type 5A is an autotransporter, indicating that the C terminus of the protein forms the outer membrane channel cylinder, right? So, whereas N-terminal uh, portion, that is pink, is exposed to the surface and released by proteolytic cleavage. So now I would like to give you also some examples of the activity of the exotoxins. For example, let's take a look at how diphtheria exotoxin works. You can see that diphtheria toxin consists of several subunits. It has typical AB structure. So, but A subunit consists of two subunits. When it binds to the receptor suitable for diphtheria exotoxin, and it is incorporated into the body, the receptor together with the uh, diphtheria toxin by receptor mediated endocytosis, the endosome is formed, right? So, and due to the acidification over the endosome, the A subunits will be released from the endosome into the environment. Then the active A subunits who target the elongation factor 2 ribosylation, and it prevents the protein synthesis. So it may lead to cell death. So this toxin is cytotoxin, or um, toxin that kills cells. That same, the diphtheриal exotoxin. Now the mode of action of Vibrio cholera exotoxin. If you take a look, it also has a B structure. So, and by B structure, the toxin binds with the ganglioside receptor on the surface of the lumen of the small intestinal tract, and also will be incorporated by receptor-mediated endocytosis, and then it will be there proteolytically cleaved into A1 and 2, and A active subunit will bind with G protein and involve in the activity of the adenylyl cyclase that cause permanent activation of CAMP in epithelial cells of the small intestinal tract, and then as a result the release of chlorine and ions outside the uh, cell into the lumen of the gut, and some other ions, so that causes the formation of losing of water and formation of the uh, rice water stool, it is called, so very severe diarrhea. So then this diarrhea, this loss of water may cause hypovolemic shock that can be a cause of the death of the patient with cholera. So, and this toxin is called so choleragen because it really generates all symptoms that we may see in cholera disease. That's also Vibrio cholera. So now the mode of action of one more toxin that is called uh, tetanotoxin produced by Clostridium tetani. Uh, so tetanospasmin, in fact, right? So how does it work? Clostridium tetani releases tetanospasmin and tetanospasmin uh, causes the continuous stimulation by excitatory transmitter and inhibit transmitter gamma aminobutyric acid or glycine release from the postsynaptic membrane, so it prevents the stage of relaxation for the muscles. We may have permanent muscle contraction, spasm, right? That you studied in anatomy department. So, and a counterpart of the tetanospasmin is the exotoxin of Clostridium botulinum. So when botulinum toxin is produced, it prevents the release of acetylcholine from acetyl-containing vesicles, right? As soon as acetylcholine is not released, the receptors are not occupied on the motor and plate, right? And then stimulation of the muscles is blocked. So we have flaccid paralysis of the muscles. And as a result, so when diaphragmal muscles also will be paralyzed, so there will be no activity in the respiratory muscles. So the patient may die out of asphyxia. So there's also description in more modern way of botulinum toxin. So my dears, we have discussed some important pathogenic properties of bacteria and mentioned some even virulence factors. Now we come up to the some epidemiological part of our lecture when we discuss how diseases can be transmitted, what can be a source of infection, and that's also very important issue. So 1 minute, I just need some water and we go on. So guys, now we take a look at the source of infection. So of course the very first one are human beings. But then the parasite may originate from two categories of patients. of human beings, the patients or carriers. I think this is understandable who are patients, those that develop the disease. But what are carriers? These are those people that are infected but don't produce any visible symptoms of the disease. But because they are infected, the microbes multiply in their, for example, oral cavity and can be excreted and may infect other people. For some diseases, the carriers make a very important group of those who may be a source of infection because nobody knows who are carriers, whether they are infected or not. For example, this is a typical way to spread typhoid fever. Usually, the carriers spread the typhoid fever because they carry the Salmonella bacilli in the gallbladder and excrete them with the stool. So, the mode of transmission of infectious disease caused by such microorganisms is called human to human. Also, or these diseases can be referred to as anthropootic infections. Then sometimes the animals may be a source of infection. Infectious diseases transduced from animals to humans are called zoonotic infections from the zoo, right? So, quite often animals may serve as a reservoir of human infections when maintain the parasite in nature. For example, I would like to give the example of zoonotic diseases. This is, for example, plague caused by Yersinia pestis. Normally, it occurs in wild or domestic animals and spread within animals, but also can be transmitted to humans. So then infection may be solved or saved in arthropods. So that's quite often. It may happen with both bacteria and viruses, right? So then this disease will be called arthropod-borne disease because these pathogens may be transmitted to humans through the blood-sucking insects like mosquitoes, ticks, mites, lice, fleas, flies. So generally all those arthropods are called vectors. We go ahead with the source of infection. Sometimes pathogens are able to survive not just in the human or animal body, but also in the soil or water for a long period of time. So, for example, Clostridium tetani may survive in the form of spores uh, in the surface layer of the soil for several decades. So then, soil or water may act as the source of infection due to contamination with such pathogens as Vibrio cholera. So, usually, the diseases caused by the pathogens that are able to live for a very long while in the environmental sources are called uh, sapronotic infection. You see, we studied anthroponotic infection, zoonotic infection, and sapronotic infection. So quite often some bacteria may survive in food. So for example, we may have food poisoning by Staphylococcus aureus as a very common cause of a common disease. So now we take a look at the stages of infectious disease. We start with typical acute disease. So what does it mean, acute disease? We have two types, acute or chronic, right? Yeah, do we have, okay, acute disease. So acute disease, I think is better. So we divide the infectious disease into several stages. First stage is incubation period. What is it? It is a period in between the entry of the microorganism inside the body and the moment when the clinical symptoms may appear. It means within the incubation period, the disease is not visible. There will be no symptoms. But then with this moment, microbes multiply already, right? So quite often, person may start to be infectious within the end of the incubation period. Generally, we consider that in the incubation period, the person is not highly infectious. But for long-term diseases like hepatitis B infection or hepatitis A infection, the person in the last term of the incubation period start to be highly infectious. Then the prodromal period uh, occur. This is a period when some non-specific general symptoms may already appear. So like tiredness or loss of appetite or headache, these are non-specific symptoms. And prodromal period is usually not so long. And in some acute diseases, it can be missed even, right? And then we have the invasive state where we have the increasing level of the symptoms, and within this term the most severe signs and symptoms of the disease occur. Then ACMAR, this is the middle of the disease, and the stage of decline, the so-called decline phase, where we see decline of symptoms and signs of the disease. And then we may see clinical threshold, the convalescent stage. We call it convalescent patients within this stage, where patients recover. But as we mentioned, recovery is very optimistic prognosis of the disease. So the disease may lead to the death. So then there will be no convalescent period, no recovery. So then, because it may go like this, right? Okay, and then the something in between is chronization of the disease. Clinical process may occur chronically. it means it will be not very severe. The symptoms will be not very severe. We have decline of these symptoms. And then they may disappear and reappear again. So, by the way, disease may go subclinically. What does it mean? So, it means we may not see clear the typical symptoms of the disease. So then, I think we've discussed portal at entry and exit already. Then, how the mode of transmission may occur. Again, I would like to demonstrate this. So first, the commonest way is a contact. But then the contact can be direct or indirect. See here, this is direct contact. Shaking hands is a direct contact. Kissing and sexual contact is still a direct contact. Что там у тебя происходит? Кого? Телефон. Ты закончил уже? Да. Ага. So then we may see indirect contact. This is a method of transmission of infection through the agency of different fomites. Clothing, towels, pencils, toys for children is very common. So the fomites may be contaminated by a pathogen from one person and act as a vehicle for the pathogen transmission to another person. So then, ingestion of food or drinks contaminated by pathogen and drinks may cause so-called intestinal infection. And they can be waterborne like cholera or foodborne like staphylococcal food poisoning or handborne or flyborne as dysentery. So then inhalation is a very common way of transmission of so-called respiratory infections or, for example, influenza or tuberculosis. Pathogens resistant to drying may remain viable in the secretion for a long period of time. So, for example, Mycobacteria tuberculosis, the common cause of tuberculosis, stay viable on the surface of the dust particles for about a half of the year. So then the disease may be also finger-borne. Transmission may be recognized in the cases of pathogen entering either through the mouth or through the nose or eyes. So insects may act as vectors of infectious disease. So these are vector-borne diseases. So, the very common way of transmission is also inoculation, because some pathogens may inoculate it directly into the tissues of the host and can cause infectious disease. For example, the spores of Clostridium tetani can be implanted in the depth of wounds. The pathogens inoculation may cause the so-called hydrogenic or nosocomial infection with the use of the non-sterile medical equipment, syringes, surgical instruments, and you should take care of this. Or it can be performed with the transmission of non-screened blood from the infected donors, and this is hepatitis B virus infection or HIV. So, by now we were talking about vertical transmission from person to person, but the pathogens can be also transmitted vertically from mother to child. This is called congenital transmission. And this is known as ability of some pathogens to cross the placenta barrier and infect the fetus in uterus. These may result in the abortion, miscarriage, stillbirth, and live infants may be born with the manifestation of the disease. For example, we may distinguish Congenital rubella, this is a viral disease. So congenital syphilis, this is a bacterial disease. And quite often, these pathogens may cause so-called teratogenic effects, such as intrauterine infection with the rubella virus, may influence organogenesis and lead to congenital malformations. So you can see here this mode of transmission. For example, do you remember Zika virus, right? So Zika infection is a vector disease transmitted by the vector, but if it occurs with the first trimester of the pregnancy, Zika virus may enter through the placenta to the fetus body, and it may cause um, microcephaly, intrauterine growth restriction, ocular abnormalities, fetal demise, long-term outcome unknowns, right? So that is very severe infection. Now we also may classify infections based on the number and order of pathogen that enter the body. For example, primary infection. This is an initial infection with a parasite in a previously healthy host. Reinfection in a subsequent infection by the same parasite in the host. Then we may distinguish secondary infection, a new infection with a new parasite, number two parasite in a host that has already infection with the primary parasite number one, and this gives some pre uh, the um, stimulating factors for parasite number two. Then we may have mixed or cross infection when a new infection in a patient already suffering from a disease caused by primary parasite. For example, if the person may have hepatitis B infection already and then hepatitis D infection also microbe enters the body. So both parasites will or both parasites may infect simultaneously. It will be co-infection. Then infections may be classified depending on the source. So it can be endogenous infection taken from outside, endogenous from host's own body, or exogenous from external source. The external are most common, of course. Also, we may classify them according to the localization of the process. For example, this is a local disease. When bacteria start producing toxin that release into the bloodstream, but the process is local, but the some toxins can spread in the whole body and we see the generalized result. So it will be focal infection. But then bacteria or virus may enter the bloodstream themselves and multiply, germinate, disseminate in the body. This disease will be called generalized infection. So, and infections also can be classified according to the clinical symptoms or clinical effects of the disease. Infection can be, as we see in the chart, subclinical, when the clinical effect is not apparent, right? So, but then the usually these patients are contagious still and may serve as source of infection. And also atypical infection when the symptoms look like differently from the regular one. So then, the very important clinical subdivision of the infection is the interacute or chronic. So acute disease is characterized by sudden onset and comparatively short course of the disease. Usually short, it means less than 3 months. and usually with typical clinical manifestation of the disease. Here we can see suspected in Ebola disease. So then chronic disease develops eventually more slow. The usually symptoms are not very severe, but then instead it lasts for much longer period of time. If more than 6 months, then this is usually chronic disease. And in different stages of chronic disease, symptoms may present or not, and usually within the chronic disease, we may have some relapses of the disease. So, the important to understand what is latent disease. So, this is the one during which the causative engine for long period may not multiply in the body, and that's why may not be found in the body, in organs or tissues. And of course, usually the symptoms will not be visible with this type of the disease, or quite often the latent stage can be one of the stages of the disease. For example, in syphilis, that is a typical stage in between secondary and tertiary stages of syphilis. Then we mentioned carrier state, right? Uh, and the carrier state may be classified into acute if occur for less than 6 or 3 months, or chronic when occur in the longer time. Okay, my dears, finally, that's good that we cover all problems that we were going to discuss today. And the class of infectious disease will be really the last class in our general microbiology course. So then, before you prepare for the class, please don't forget to read lecture. Okay, my dears, goodbye. The next lecture will occur on the 16th of May. And then we start already special microbiology lectures. Goodbye.